Why do you think the cell spends so much time and energy, remember GTP, in this process of folding? Why would a cell um, target very particular processes like the unfolded protein response and the ERAD response towards repairing misfolded proteins? What is it about folding that's so important? Remember, the shape that a protein takes, its folded shape, is going to determine its function. And we know from discussions in this class that even one change, one additional molecule being added to a protein, one molecule being taken away, any mutation in the DNA that causes even one amino acid difference, all of those things can cause a protein to change shape significantly enough to change its function. So that's certainly one reason that the cell devotes so much time and energy into it. The other thing I want you to think about, though, is the fact that sometimes when a protein folds differently, abnormally, the new function that it takes on can actually be quite damaging to the cell. Sometimes a misfolded protein just doesn't work. Other times, the misfolded protein starts doing a function that's actually damaging to the cell. In the image that you can see on this slide, on the left-hand side, you can see a folded protein. You can see that there's some green alpha helices drawn, there's some blue beta-pleated sheets drawn, and then there are some yellow loops of short sequences of amino acids. Now compare that to what's sitting next to it on the right-hand side. You can see that instead of having, I guess, 3 alpha helices, now there are only 2. And instead of having 1 or maybe 2 beta sheets, now there are several. This protein has folded in a different way, and that ends up giving it a different function. And in this case, that function happens to be disease. The protein on the left-hand side is perfectly normal. The misfolded protein on the right-hand side is what we call a prion. Prions are proteins that can cause very serious neurologic disease in certain organisms, including humans. I'm sure you've heard of mad cow disease. Mad cow disease is a process where a cow slowly starts to become unable to perform any normal neurological processes. The cow eventually cannot stand, they cannot walk, they start to have seizures, they go blind, and they die. There are human forms of mad cow disease. There's one called Creutzfeldt-Jakob disease. There are also other forms of prion diseases in other species. Sheep, for example, get a prion disease called scrapie. In fact, many other mammal species have their own particular prion disease. The other really scary thing, as you probably know, is that the prion diseases can be passed from species to species to species. We know, for example, from past outbreaks that if a human eats any nervous tissue from a mad cow infected cow, that human can then develop the prion disease themselves. Prion diseases can take a whole long time to show clinical signs, which also makes them very scary. People can eat a hamburger that comes from a mad cow disease cow and not develop clinical signs themselves for up to 10 years later. So prion diseases are devastating, and they're not caused by infectious organisms in the traditional sense. They're not caused by bacteria or viruses. They're caused by misfolded proteins. All right, the diagram on this slide kind of puts together everything we've been talking about so far. You can see in the upper left-hand corner the beginnings of this pathway that many proteins take through co-translational import. We can see the mRNA leaving a nuclear pore, and then you can see where the arrows are showing us that that mRNA and its attached ribosome are eventually headed for the co-translational import process. If you follow the arrow, you can see that little tiny boxed region blown up so that we can see it. And it's showing us that the ribosome has bound to the ER membrane, the translocon pore has opened, and translation is allowing the polypeptide to be formed inside the lumen of the ER. Now, if you go back to the larger diagram, we can see a vesicle budding off of the ER with the protein in it. We can see that vesicle traveling to the Golgi apparatus and merging with the cis face. The protein's going to have further modifications in there. It's going to exit through the trans face. And then we see that there's all kinds of places that that vesicle can go. For example, the vesicle could carry the protein to the membrane and allow it to be released to the outside of the cell. Or the vesicle could end up going to any of the other destinations that we know are involved in co-translational import. For example, the protein could be carried back to the RER, or carried back to the Golgi, or it might be sent off to a lysosome, or it might become part of the transport vesicles. Any of several different destinations are possible. Okay, so that finishes our description of co-translational import into the ER lumen. Remember that for some destinations, it's a post-translational process. We're not going to talk about the post-translational process in any detail, but I do want you to understand a couple of things. First of all, in the case of post-translational, the reason it gets that name is because the proteins will be imported into their destination after translation has occurred on free ribosomes in the cytosol. After translation, those proteins will then be taken to their final destination. For some of them, that means remaining dissolved in the cytosol itself. For some of them, that means being imported into one of the final locations we discussed, including the nucleus, the mitochondria, chloroplasts, or peroxisomes. Now, I want to go back to co-translational import for a few minutes because there's a particular type of protein that I want to discuss in a little more detail. And that's proteins that are destined to become part of the plasma membrane. When it comes to membrane proteins, it doesn't matter what kind of protein we're talking about. Any protein that's destined to become part of the plasma membrane is going to undergo co-translational import first. So remember, that means it's going to be imported into the ER lumen, and then it's going to be processed, including things like cleavage and splicing. It's going to go through its chemical modifications. It's going to be folded, and then it's going to be trafficked to its ultimate destination, which again is the plasma membrane itself. Now I have a little note here with a couple of asterisks. In addition to the plasma membrane, this is the same kind of a process that would happen for a protein that's destined to reside in the outer organelle membrane. So it's important to remember that all membrane proteins, all proteins that are destined to live in either the plasma membrane or an outer organelle membrane, are going to first be embedded into the ER membrane. So they have to go into the ER through co-translational import, and then they are literally going to be inserted into a portion of the ER membrane. So integral membrane proteins, IMPs on this slide, are going to be incorporated into the ER membrane, and then that little piece of membrane with the IMP in it is going to bud off of the ER into a transport vesicle. And that transport vesicle will be sent to either the plasma membrane or some outer organelle membrane, where it will fuse with that membrane, and the IMP will be in place. Just a couple of reminders about membrane proteins. When we talk about integral membrane proteins, or IMPs, we're talking about proteins that are inserted in the membrane. But it turns out that there are slight variations among IMPs. The ones that most of us are comfortable with are transmembrane proteins. These are proteins that get inserted in the membrane all the way across. So they span the entire membrane, both monolayers. Less commonly, you can see what are called integral monotopic proteins. And what these are proteins that pass through the membrane, but only on one monolayer. So they go through one monolayer only, and they emerge from the membrane, therefore, on only one side. Now, integral membrane proteins of either the transmembrane type or the less common integral monotopic type are usually held in place, and I mean that relatively speaking. They're usually kept within the membrane because they have alpha helical segments in them that are hydrophobic. They contain somewhere between 20 and to 30 hydrophobic amino acids, which can interact with and remain within the tail section of the membrane. Now remember, most proteins can move around in the membrane. They can go through a diffusion process in the membrane. That's part of our fluid mosaic model. But the reason that they can even sit in the membrane is because they have a region of hydrophobic amino acids that curl up into an alpha helix and comfortably sit in that hydrophobic tail region. The other thing to remember is that transmembrane proteins can pass through the membrane, one side to the other, one time or many times. So in other words, some proteins pass through the membrane and that's it. They are called single-pass proteins. Other proteins are so long and complex that they're capable of passing through the membrane, turning, passing through the membrane again, turning, passing through the membrane again, and so on. Those are called multipass proteins. This is a picture that demonstrates for us how membrane proteins differ. Remember, some membrane proteins are only going to associate with the polar head region of the membrane. We call those peripheral proteins. And you can see some on the top left part of the slide. You can see a pink peripheral protein and an orange peripheral protein. They are composed almost entirely of polar amino acids. In the center of the slide, you can see a blue protein that has some regions that are outside the membrane, but also several helical regions that pass through the membrane. That is a true integral membrane protein, a transmembrane protein. It crosses all the way through the membrane multiple times. There's a purple protein, another transmembrane protein that's a single pass protein. It's also got a peripheral protein in pink associated with it. Now, if you look at on the lower part of the slide, on the left-hand side, you can see a green protein that part of it passes through one of the monolayers. That would be one of those unusual integral monotopic proteins. All right, so we know that all membrane proteins, integral membrane proteins, are originally inserted into membrane in the ER, and that piece of membrane with the protein in it has to then be packed into a vesicle and taken to its final destination. So the question is, what marks a protein as being an integral membrane protein? And the answer is that there's going to be some particular sequence of amino acids in that protein that marks it as needing insertion into the ER membrane. Now, some IMPs have a signal sequence very similar to the ER signal sequence. But rather than saying that all IMPs have a signal sequence, we have to be careful, because some IMPs have a particular signal that's not at the N terminus. There are some IMPs that have very specific amino acid signals in other locations. So we'll think first about a case where there's a series of amino acids within the protein that we're going to call a stop transfer sequence. Okay? This is the type of signal that marks a protein for insertion into the membrane, a stop transfer sequence. And you'll see in a minute why it gets its name. What this sequence of amino acids has in terms of chemistry is that its amino acids are all hydrophobic. Any protein that has a stop transfer sequence within it also has an ER signal sequence at its N-terminus, because of course it has to get sent into the ER first in order to be integrated into the ER membrane. So the ER signal sequence, as it always does, makes sure that that protein gets co-translationally imported into the ER. It interacts with the SRP, which connects the ER signal sequence with the translocon, and it proceeds just like any co-translational import. But the difference is right here. As that protein gets translated, translation is going to continue with the growing polypeptide passing through the pore and emerging inside the lumen until that stop transfer sequence gets translated. That hydrophobic region gets translated. At that point, what's going to happen is those amino acids are going to be strongly attracted to the tail region of the membrane. Remember, this protein is being co-translationally imported. That pore is passing right through the ER membrane. So translation will keep going. The ribosome will continue to translate the mRNA, creating this new protein. But translocation is going to stop. In other words, the process where this growing protein is passing through the pore into the ER lumen, it's going to stop. And that's going to allow the protein to continue to get bigger and bigger, but stay, for the most part, out in the cytosol. Let me show you what I mean. Take a look at this image. In the upper left-hand corner, you can see only a part of the machinery that allows co-translational import to occur. You can't see the ribosome, you can't see the mRNA, you can't see the SRP or even most of the translocon. You can see a piece of the ER membrane, and you can see in a light pinkish purple color the pore of the translocon. The green region is the ER signal sequence, so that's inside the pore, bound to the wall of the pore. You can see a purple strand of amino acids, a polypeptide, passing through the pore from the outside of the cell as it's being translated. As you move to the center image up top, you can see that the protein is growing longer and longer. But now we can see this red sequence. That's the stop transfer sequence. When it gets translated, as it passes through the pore, it's going to stop. It's hydrophobic, and it's going to be attracted to the tail region. So translation is going to keep happening, but translocation is going to stop. So any of the protein that's already in the ER lumen is there. But any of the rest of the protein that gets translated is going to be stuck outside the cell in the cytoplasm. And that's what they're showing you in the third diagram up top from the left. Most of that final protein, the C-terminus, in other words, is outside the cell. A small portion of it is inside the cell. And it's a transmembrane protein. It crosses all the way through. So by putting a stop transfer sequence inside this sequence of amino acids, what the cell has achieved is the creation of an integral membrane protein that is literally stuck in the ER membrane. That thing can then be packaged into a vesicle and sent off to its final destination. Now you'll notice up top, third diagram over from the left, that this integral membrane protein, newly created, has left the pore. See how it's no longer sitting inside the pore? Remember, that's because hydrophobic amino acids can transfer themselves, can diffuse laterally in the membrane. And that's what this one has done. Now take a look down at the bottom. Something very different is going on down there. So if we want to create an integral membrane protein, we're first going to put it into a piece of membrane in the ER. Okay? And for some proteins, we're going to end up with a protein that has most of its bulk outside the cell at the C-terminus side, and a small amount of it at the N-terminus side inside the lumen. It's also possible, though, to create an integral membrane protein inside the ER membrane that is reversed from that. In other words, it's possible to have a protein embed in the ER membrane where most of the protein ends up at the N-terminus and only a small amount at the C-terminus. These proteins are structurally quite different. They don't have an ER signal sequence. Instead, they have what's called an internal start transfer sequence that looks almost like the ER signal sequence. So it has a very similar sequence of amino acids, but rather than being at the N-terminus of the protein, it's internal. It's located farther up inside the sequence of amino acids. What's going to happen is the SRP is still going to recognize it, and it's going to take it and allow it to bind to the translocon. But it's going to bind very differently because, again, we're not at the N-terminus here. We're at some internal part. So if I go back to the last slide, you can see something very different happening down at the bottom. If you go over to the left side, in the bottom part of the diagram, you can see that there is no green region at the N-terminus of this purple protein. The green region is farther in, and we're going to call it an internal start transfer sequence. The SRP is going to guide that green region to the pore and help it bind to the wall. That's going to leave the N-terminal part of the protein outside. As translation starts up again, and the protein starts to feed through the pore, eventually the entire protein will be translated, and most of it will be inside the cell this time. So if you compare the last picture from the top three to the last picture in the bottom three, you have two integral membrane proteins. But in the top one, the C-terminus is a much larger portion of the protein than the N-terminus. And on the bottom, the orientation of that has been reversed. The bulk of the protein at the C-terminus is now inside the cell, and the smaller amount of the protein at the N-terminus is now outside of the cell. So it turns out that when a cell embeds a membrane, embeds a protein into the ER membrane to create an integral membrane protein, it all depends on where it needs this protein to sit, how it needs it to orient. Translation can occur in a way so that most of the protein ends up in the cytoplasm, or it can occur in a way so that most of the protein ends up outside the cell. And it all depends on whether or not the protein has an ER signal sequence on it and a stop transfer sequence, or just an internal start transfer sequence. That's going to determine how the protein ends up in terms of its orientation. So if you look at the picture on this slide, again, we see on the left-hand side, down towards the bottom, we see this protein which has started to be translated out in the cytoplasm, and we see a blue translocon pore just underneath it. Notice the red sequence. That's a start transfer sequence. The SRP is going to bind to that start transfer sequence and take it down to the pore and help it bind inside the pore. Then translation is going to begin. as more and more and more of the green protein gets translated, it'll feed down through the pore. The start transfer sequence is keeping it locked against the wall of the pore. So once translation is complete, most of that protein ends up inside the cell in the cytoplasm of the ER lumen, and a small amount of the protein ends up outside of the cell, just in the general cytoplasm, the general cytosol. So both of those descriptions, the description with the ER signal sequence and the stop transfer sequence, and then the description of proteins that don't have an ER signal sequence but have a start transfer sequence, both of those are going to end up creating integral membrane proteins that are single pass. But what about multi-pass proteins? How do we get a protein to insert itself across the membrane several times? We're not going to get into this in any detail, but just to finish up today, I want you to know that the way the cell can achieve this is by placing alternating stop transfer and start transfer signals inside of a protein's amino acid sequence. So if you look at the picture on this slide, again, the area at the top half of the slide represents the general cytoplasm, and the area on the bottom part of the slide represents the ER lumen, the fluid inside the ER lumen. So we have a gray stretch of membrane, we have a blue pore that would be sitting in the translocon. So the green protein that has started to be translated out in the cytoplasm is what you can see over on the left-hand side. There's a red start transfer sequence, and there's a yellow stop transfer sequence. There's no ER signal sequence. The SRP particle, which you can't see, is going to bind to and guide that red region down to the pore. It's going to help that red region bind to the wall of the pore. When translation starts up again, more and more of that green protein is going to enter down through the pore into the ER lumen until that yellow region gets through the pore. Remember, the stop transfer sequence is also hydrophobic. So that yellow region is going to stop in the pore and bind to the wall. Okay? Now, this is a simple example, but at that point, that's a multi-pass protein. It will go through lateral diffusion to get out of the pore, and as you can see on the right-hand side, it has passed the membrane twice. Once, one pass was achieved with a start transfer sequence, and one pass was achieved with a stop transfer sequence. And you can imagine that if you added more of these, if you kept alternating start, stop, start, stop, start, stop, you could get a protein to pass through multiple times.